All right, our next speaker is Joe Takahashi. So now we're back to people who have been with the Institute since the very, very early days. Joe was on the original charrettes, which were for planning the whole Allen Institute for Brain Science and the Allen Brain Atlas project, and was on our scientific advisory board for many years, helping us all along the way. And it's a great pleasure to welcome Joe.
Thank you very much, Alan. And I wanted to especially thank Paul Allen for his vision in creating this institute. I recall from, let me just type in my password here. My first recollection of the Allen Institute is I received a phone call from David Anderson, who said, Joe, I have this really interesting activity that I think we would like you to join, but I can't tell you anything about it until you sign some paperwork from some company called Vulcan. So I said, well, you know, David Anderson says it's interesting, I better do this. So I signed the paperwork, and of course, it was to go to the first charrette. I want to thank Jim Watson and David for inviting me to that. It was an incredible event, and as you can see today, the results of that charrette were just incredible. And I really think that all of us owe great debt to Paul Allen and Jody for supporting this, and for the original people who conceived of this idea and making the Brain Atlas a reality. So my role, I think, in the early days was because I'm really a mouse geneticist. And in addition to looking at genes in the mouse, Honghui Zheng was eventually recruited. And as you know, many tools have been developed here using gene targeting, which have been a great boon to the entire field. So thank you, Honghui. So today, what I want to do is to briefly tell you a story about the molecular basis of clocks in mammals. And in humans, light is the major synchronizer of our clock. There are a special set of ganglion cells that project into the hypothalamus, into these two yellow wing-like structures, which are called the suprachiasmatic nuclei. And within each nucleus, there are about 10,000 neurons, each of which can generate circadian rhythms of electrical activity, as shown by David Walsh in 1995. But today, what we know, and that's the theme of today's talk, is that really circadian rhythms are a property of cells. The fundamental unit of the clock is a cell-autonomous molecular clock. And at the core of this clock is a set of 6 transcriptional regulators, 2 activators, clock and BMAL, which are BHLH pass proteins, that regulate 2 principal repressor proteins, PER1 and PER2, Cry1 and Cry2. These are transcribed. The RNAs are translated, the proteins accumulate in the daytime, they physically interact with each other, and then at night they translocate back into the nucleus, where they physically interact with clock and BMAL to repress their own transcription. So this is what we call the core molecular feedback loop of the mammalian clock system. And the way that we study this system is to use reporter gene technology. This is an example of a luciferase fusion to the endogenous PER gene constructed by gene replacement. This allows us to visualize with bioluminescence the abundance of the PER2 protein. And this movie on the left, which is a 7-day time-lapse recording done by David Walsh, who's now at UCSD, is an organotypic slice through the hypothalamic suprachiasmatic nucleus. And we can visualize, these are the 2 nuclei here, we can visualize per gene expression over time. And as you can see, it's oscillating. It's high in the daytime, low at night. And there's also a very stereotyped wave pattern that goes across the nucleus. Okay, so this is one of the tools we use. And with this kind of mouse, we found that many cells in the body could express circadian rhythms. So here's an example of primary fibroblasts made from skin from these mice in a monolayer culture. And this is, again, a recording done by David Welsh. But it's an incredible experiment because it lasted 6 weeks undisturbed. And what you can see here are these little twinkling stars. Each of this is a fibroblast with PER2 expression oscillating. And on the right, I've shown you 6 examples to show you the PER2 oscillations that are persisting for 42 days in culture. It's incredible. Each cell has its own periodicity. There's no coupling among the population. And so this experiment fundamentally changed our view of the system. Before, we thought the clock was in the brain. The brain, the SCN, was in charge, the pacemaker, and it sent signals throughout the body, and the body was a passive follower of circadian rhythms. But with the cloning of genes, we discovered that the genes are ubiquitous. Almost all cells express them. And today, we now know that essentially every cell in the body has the capability of expressing circadian rhythms. And so the brain is still in charge, but all the major organ systems have cell-autonomous local circadian oscillators that generate molecular oscillations in those tissues. And so this has raised a number of questions. What's the organization of the system? How does the SCN talk to the liver? Is the SCN still in charge, or does the liver do whatever it wants to do? And what are those signals? So today, I'm not going to talk about any of those organizational questions. I want to go back to this core feedback loop and then really focus in on one aspect of clock B malfunction. First, we are beginning to make some inroads into the atomic structure of some of these components. So this is just a movie showing you the crystal structure of clock B-mal, which are BHLH pass proteins that we presented last year. I'm not going to talk about that, but we hope this is really the beginning of a molecular understanding of how these 6 proteins might function to constitute a clock system. So instead, what I want to do is to focus on what clock and B-mal are doing in the genome, because they're transcription factors. And as we've heard from Richard Gibbs, sequencing technology has really opened up a revolution in our ability to study various processes in the genome. And one of these is to be able to identify the binding sites, or the target genes of transcription factors, using a technique called immunoprecipitation followed by next-generation sequencing, or ChIP-seq. So what this involves is to immunoprecipitate chromatin with specific antibodies to, say, clock or B-mal, purify that DNA, and then sequence it to map the locations of where the protein was binding. And so using this kind of technique, this is what the data would look like for a single gene. This is B-mal DNA binding at a single target gene called DBP, which is very well known. There are 3 binding sites, one in the promoter, one in the first intron, and one in the second intron. And each of these blue profiles shows the binding sites at 6 different times of day, 0, 4, 8, 12, 16, 20. 0 is dawn, and 12 is dusk, or light set. And so what you can see is binding is high between 0 and 12, that's the daytime, and then low at 16 and 20 at night. And then KO is a knockout mouse control for B-mal. That's to show the antibody specific. There should be no signal. Okay? So that's just one factor, B-mel, one gene, 3 sites. What about the whole set of transcriptional regulators? So there are 3 activators, B-mel, clock, and NPAS2. NPAS2 is the paralog of clock and can substitute for the function of clock. They both NPAS2 and clock interact with B-mel. They're activators. They have the same pattern. They bind the same sites. They have the same temporal pattern. They bind in the daytime. Then the 4 repressors, PER1, PER2, Cry1, and Cry2, bind the same sites, primarily in the promoter, but they have a different temporal pattern. They're high at night. They come on at 12 to 16:20 in the case of PER1, PER2, and Cry2. And then Cry1 has a third pattern. It comes on late, peaks at 0 at dawn, and then fades away. I'll come back to say what that might mean. OK, so this is all still one gene as a target for now 7 factors. What about the entire genome? So this is a view of the whole genome for B-mel and clock represented as heat maps for binding intensity at the 6 times of day. So now there are 5,900 sites oriented vertically, with red being high binding. And what you can see is that B-mel binds with a peak, say, around 8, 4 and 8, across the entire genome. It's low at 16:20, no signal in knockout. Clock also peaks at 8. And then PER1, PER2, and Cry2 have this nocturnal pattern. And then Cry1 has a third pattern, which turns out to be a mixture of a 12-hour and a 24-hour pattern. Okay, so it looks a little complex here. The other surprise is the number of binding sites. So we suspected that clock and B-mel might engage perhaps thousands of genes. But this shows that they engage at least 3,000 genes directly in the liver. But the real surprise are the cryptochrome proteins, which have many more thousands of binding sites, 16,000 in the case of Cry1, interacting with perhaps 6,000 genes in the genome. That's half of the genes that are expressed in the liver. Okay? What about the sites of interaction? So this is a 6-way Venn diagram looking at the overlap of clock, B-mel, and the 4 repressors, shown in the different colors. In the middle are 1,400 sites where all 6 factors bind exactly the same site. And then around the periphery are all the other combinations, 5 out of 6, 4 out of 6, 3 out of 6, 2 out of 6. And then here are sites that are unique, Cry only, Cry1 only, Cry2 only, many thousands of them. And this is interesting because these are sites where Cry is interacting with DNA, but there's no clock B-mel. And we have validated this. This is true. And this is consistent with reports that Cry1 can interact with other transcription factors, such as the glucocorticoid receptor and other nuclear receptors, such as reverb alpha. OK. So this is just occupancy. What about function? So to look at function, we use RNA-seq to look at transcriptional readouts across the whole genome as a function of time of day. And so this is a browser view of the PER2 gene, which is on the minus strand, so it's going from right to left. It's a larger gene. It's about 50 kilobases in size, has 23 exons, compared to DBP, which is only 2 kb in size. And here we have 12 samples taken every 4 hours over 2 days in constant darkness from the liver of a mouse. And I forgot to mention that the liver is the major target gene for the brain as far as clock outputs, and is currently our model for the brain because it's a homogeneous tissue. So here in black are shown the sense reads from the PER2 gene. If you look at the 3' UTR, you can see it's low here, high here, low here, high. And it has a very nice rhythm shown in this graph here. And this sequencing information is strand-specific. So with this, we discovered a new transcript in the per gene, shown in red, that's antisense. It begins in the middle of the gene, goes the other way. Interestingly, this one is also oscillating. It's antisense, antiphase. And we're studying the function of it. It might be regulatory for the main transcript. We don't know yet. And then finally, we can annotate these data in different ways. We can look at the transcripts in the whole gene body, or just in the introns, or just in the exons. And we looked at the intron signal because we use it as a proxy for pre-messenger RNA levels, or that would be a proxy for nascent transcription. Okay? And then we can use the exon signal as a proxy for mRNA, or mature or processed RNA. So when we annotate genes this way and look at whether they cycle, we find about almost 1,400 genes that have cycling intron transcripts. So these presumably are transcriptionally regulated by the clock. And then over 2,000 genes that have exon cycling transcripts. The surprise came, though, when we looked at the timing of these rhythms. The intron transcripts are highly synchronized in the genome with a single peak at CT15. That's 3 hours into the night, or the active phase of the mouse. Whereas the exon transcripts are more bimodal and spread out. This is what we saw with microarray 10 years ago. Okay? And then if we compare these two gene sets, there's surprisingly a little overlap, as if many genes might not be driven transcriptionally, or we couldn't detect them in this level of analysis. But our interest really focused on this new synchronized burst of transcription, and that motivated us to look at RNA polymerase II, the machine that actually transcribes RNA. And so this is just an illustration of the early steps in transcription from Jerry Workman's lab. The first step are the binding of activators such as clock and BMAL to regulatory sites in genes, which then recruit chromatin modulators such as P300 and CBP, which are histone acetyltransferases to kind of open up the chromatin. And then this allows recruitment of the pre-initiation complex, shown here. And then one of the first steps in initiation is serine 5 phosphorylation on the C-terminal domain of the large subunit of polymerase II to initiate transcription. And then there's a second phosphorylation on serine 2 for elongation. So we use ChIP-seq to look at RNA polymerase II, and this is the PER1 gene in this case, shown here. There's the promoter. Here's a BMAL at the promoter. And what we see here is hypophosphorylated Pol2. This is a newly recruited form of Pol2. And we find that its occupancy is highly circadian. There's a very strong peak at 12, which is the maximum amount of transcription for this particular gene. And then the serine 5 version is also rhythmic, but the peak is much broader. Okay? But the real surprise is when we look genome-wide in the liver. So this is all sites in the liver, about 7,000 sites. Pol2 occupancy is circadian across the entire genome. That was a complete surprise to us, whether or not those genes have cycling RNAs or not. And it's synchronized. It peaks at CT14.5, which is just a half an hour before that nascent transcriptional burst. So we think that that might be underlying this synchronized burst of transcription. When we looked at serine 5 occupancy, we got a second surprise. It is also circadian, but the phase is totally different. It peaks at CT0, and there are many more sites. There are actually more than 20,000 sites in the genome where serine 5 occupancy is circadian, and 13,000 of those are significant for circadian cycling. Now 0 is interesting because that's where Cry1 peaked, and we can see very significant overlap between serine 5 and Cry1 occupancy, many genes. And in fact, we can see a complex of clock BML bound by Cry1 with serine 5 Pol2, suggesting that clock BML can recruit Pol2 initiated, so it gets a serine 5 mark, but then it pauses or remains in a paused or poised state. And I'll come back to what that might mean. So given these changes in RNA polymerase II, we then were motivated to look at changes in chromatin state, which are largely either a consequence or a predecessor of transcription in many systems. And so histone methylation and acetylation are very characteristic marks of different steps in transcription. So here's a browser view of the PER1 gene again. Here's the promoter. There's BML to orient you. And what we found was really interesting. Histone 3 lysine 4 trimethylation, which is a classic mark for a promoter site, is there, but it's dynamic. This is thought to be a stable mark for promoters, but it isn't. It's changing every day. At 0 and 4, it's not there. Peaks at 12 and then goes away. Lysine 9 acetylation and lysine 27 are also promoter enhancer marks. And you can see that these are also highly dynamic and circadian. Okay? And so if we then look across the genome, these are profiles. Each colors a different time for 2 forms of polymerase and these 3 histone marks I just showed you. This is for all expressed genes in the liver. When we look at the genome average, we can see that the chromatin state is actually cycling on average across the entire genome. If we look at unexpressed genes, there are no marks. We did this because in embryonic stem cells, there are marks, even though the gene's not expressed. This is not true in the liver. These are cycling genes. You can see that these also carry cycling marks. They're slightly enriched to the genome average. But the surprise is the sheer number of these genes that carry chromatin marks in the order of 4,000 to 5,000 genes, many more than we saw for RNA cycling. And then the final surprise is if we look at expressed genes that don't have detectable RNA rhythms, they're still carrying cycling polymerase and histone marks. Okay? And finally, if we then try to explain what's going on, it turns out that the best relationship between occupancy is not whether the gene is cycling or not, but rather whether it's expressed or not. So there's 90% concordance for both specific clock transcription factors and general factors such as CBP, P300, and Pol2. Okay? For expressed genes, of course, non-expressed genes have no occupancy, but cycling genes are really not especially enriched. So it's not cycling that occupancy is predicting. It's actually predicting expression. So let me summarize all these results here, trying to put all of these data together into a day in the life of the liver cell. What we find is a new state in the morning at dawn where clock and BML are bound, but there's no transcription, and that's because it's being repressed by Cry1. It can still recruit Pol2, but it's in a poised state until Cry1 levels fall. Then this begins the real activation phase where we see significant occupancy of all of the activators, recruitment of P300, and these activation marks on chromatin such as K9 acetylation, K4 monomethylation. Then we see this burst in transcription here, and then shortly after this is followed by occupancy of the repressors to inhibit the activation cycle. Then they go away, and then the cycle starts over again. So that's a very simple view of the life of a liver cell, our model of the neuron, facetiously. But I think what it illustrates is that the clock is doing much more than we thought in previous work. It is engaging this basic machine of the cell, RNA polymerase II, on a daily basis across the entire genome of the liver. And this is really a much more pervasive control mechanism than we suspected based on our previous RNA expression results that we did in the past. So I'll stop there. And the majority of this work was really done by these individuals here, Nobu Koike, in collaboration with Kim at UT Southwestern. And again, I'd like to thank the Allen Brain Institute for all of their work for the field of neuroscience. Thank you.
So I'm wondering what the signal is from the brain to the liver, and is insulin involved?
Oh, there. Thank you. So it turns out that there are many redundant signals from the brain to the liver. So Uli Schibler's lab has shown that glucocorticoids can synchronize the liver. We've shown that the subtle variation in body temperature is a very strong synchronizer of all the peripheral organs, including the liver in the mouse. And there's some serum factor that is circulating that is also another signal. The source of that is not known, but it activates an SRF signaling pathway that was recently shown by Uli Schibler's lab also in a Cell paper.
What's the average phase shift between two liver cells in terms of clock and BML1? And then B, what happens if you lesion the retinal genetically or optogenetically, or if you lesion the retinal ganglion cell input into SCN? How quickly does that phase shift drift?
Okay, so I'll answer the retinal part first. So the retinal ganglion cells express the photopigment melanopsin. If you knock out melanopsin, there's just a partial loss of function. You actually have to take out the rods and cones because those ganglion cells integrate the information from rods and cones and project them to the SCN. So if you do a triple knockout to get the rod, cone, and melanopsin, then the animal is totally blind and doesn't synchronize. And it's as if the mouse is in darkness, free runs. So those experiments are done by Samer Hattar and Sachin Panda. So as far as your second question about liver, how similar or different are liver cells? So that's an extremely interesting question. So if you look at the single cell level, like that fibroblast experiment I showed you, the periods of single cells go from 22 hours to 30 hours. And in this long recording, they're incredibly stable. They're not variable, fluctuating from 22 to 30. Each cell has a very stereotyped and characteristic period.
In a single individual?
One cell. Yeah, so this is a culture made from one mouse, in this case. We've taken this a step further. We've made this cell line immortalized. The imaging looks the same. The cells have this broad distribution. But then what we did is we made single cell clones from that parent culture. And what we find is the clones actually have unique periods that have a distribution almost as broad as the primary culture. So there's a clonal component to period determination. But then when we do single cell imaging on those clones, there's a spread. So there's both a stochastic or environmental component, and there's a clonal or genetic component that contributes to the single cell variability in period, which we're studying now. And Well, I'll stop there because it's a long answer to give you all the details.
I have a pair of questions.
Mm-hmm.
One is, why is so much of the genome involved from an evolutionary perspective, if you have an idea? And the flip side of this, as I try to wrap my mind around the results, is in blind people, are there large dysfunctions in liver that can be traced to not getting this input?
Good question. So the liver is perhaps one of the tissues that has the highest enrichment for cycling genes. And I think physiologically it does make sense. I didn't show you any of the specific annotations for those genes, but those genes are all enriched for every metabolic pathway in the cell. So, and this is true when you look at many different tissues, not only the liver. So our view today is really the clock is a housekeeping function of the cell. And it is actually engaged in regulating metabolism at all levels of the cell. Perhaps for efficiency reasons, we don't really know yet. And in the case of the liver, the fraction of the genome is so high because most of the liver is engaged in dealing with rhythmic food input. So not only processing in a metabolic way, but also detoxifying. So P450 pathway is highly circadian. We've known that for a long time. So there are many aspects of the behavior of feeding that lead to different metabolic and energetic demands for that tissue.
And the second part was about people that are blind, if there are obvious anomalies.
Yeah, so some blind people who cannot synchronize to the 24, and they have very great difficulty in dealing because they're free running. So sometimes they're in phase and other times they're out. So for those individuals, it is a serious problem. Some of those individuals are treated by melatonin, but I think the metabolism of those individuals hasn't been well studied, so we don't know if they have metabolic defects.
I think we have maybe time for one more question from
Quick question. I'm confused as to why BMAL binding is offset by almost 10 hours from the transcription elongation. Is that because this is a nocturnal species and you need the transcription at night when the animal is active and foraging? If you looked in a diurnal species, you think you'd see that the timing of BMAL binding was the same, but there wasn't this 10-hour delay, and that transcription would initiate much faster in relationship to the onset of binding?
Yeah, very good question. So no one has done that experiment in a diurnal organism, so we actually don't know the answer to that. But you're right, there really is a very long delay.
It may be that BMAL's tied to the sun, which is the same in every animal.
That's right.
But the transcription is related to metabolism, and that is on when the animal's active, and that'll be different for a nocturnal versus a diurnal animal.
Yeah, I would say there is evidence for that because it's clear that some of this rhythm that we see in the liver is driven by rhythmic feeding rather than cell autonomous clock function. So it's really, you can think of two major inputs to this gene expression pattern.
Okay, let's thank Joe again.